First of all, thanks so much to the organizers for having me here today. My name is Liam, and I'll be talking about the structural causes of pattern formation and its breakdown, and hopefully that becomes a little less vague as I get into the talk. And I am a PhD student, so this is, of course, joint work with my advisor, Dr. Adriana T. Dawes, over here, and we work at The Ohio State University. And we are interested in developmental biology, and oftentimes in development, you'll have a group of cells that has the same developmental potential. But then at some point, an external signal called an inductive signal will come in, it'll cause these cells to communicate, and then after communication, a pattern of cell fates will form. So now in this schematic, you can see the pink cells, they're on one developmental trajectory, while the white cells are on another developmental trajectory. And this process tends to repeat itself over and over again in development. So oftentimes these cells will then proliferate again. There will be more groups of cells with the same developmental potential, and more patterns of cell fates will form. So this is called sequential induction, and it's one of the most common ways that we get all the complexity, all the different cell types in the adult organism. And furthermore, if this pattern breaks down at any of these steps, it can be detrimental to the function of certain tissues. And so we really want to understand how these patterns can form and how they can break. So fortunately, there's been a lot of existing mathematical research on this topic, and people tend to study this system through modeling them with differential equations. And even though, so they mostly focus on a process called lateral inhibition, where cells are essentially inhibiting each other. They're competing for amounts of signal until some have a high amount of signal destined to become one cell type, and others have a low amount of signal destined to become another. And this most commonly occurs through a chemical signaling pathway called Notch signaling. And even though it's extremely complex, in 1996, Collier et al. Proposed that we could distill it into just keeping track of two biochemicals within each cell. So typically, we're modeling a whole array of cells, but schematically, I'm just going to represent it with two cells. So within each cell in this array, Notch is inhibiting delta, and then for cells communicating with their neighbors, delta is activating Notch. And within each cell, we can write down two equations to model the system. Typically, they'll take some form like this over here on the left. So you have the concentration of Notch, the concentration of delta. And for this talk, the details of this aren't going to be too important, but this H and G over here are typically Hill functions like Michaelis-Menten kinetics. And then you have these decay terms as well because these biochemicals degrade over time. So the really nice thing is that when you simulate these systems, you'll often see that a pattern of cell fates forms like this. So I also took this from the Collier et al. Paper, and these black cells, they have low Notch and high delta, and the white cells have the opposite, so these are the two developmental trajectories. Interestingly, they also found that these patterns tend to form through a bifurcation. And so what that means is for low amounts of external signal to the cells, you'll usually see a homogeneous steady state where all the cells are the same. But then as you increase that external signal, you get multiple steady states forming associated with the pattern. And of course, this model was first proposed about 30 years ago now, so there's been several improvements since then. And most of them tend to focus on being able to see more biologically realistic patterns. And in particular, in their original paper, they were really only able to see what are called fine-grained patterns. And so people have tried to see coarse-grained patterns that span over multiple cell widths, such as larger spots or larger stripes. And in the literature, there tend to be two ways that we can see the coarse-grained patterns. The first is that we can allow cells to communicate over longer ranges. And the second is that in addition to lateral inhibition, if we add an additional type of chemical signaling, such as something called lateral stabilization, we can also get these coarse-grained patterns. So this is all very nice, but I think one of the biggest challenges with mathematical modeling is that we do have to make all these assumptions when modeling the system. And in this case in particular, we're assuming the chemical reaction rates, we're assuming which cells are communicating, and we're assuming the chemical signaling pathways involved. And so our goal when we set out to do this project was to reduce some of these assumptions in the system. And in particular, we want to reduce the assumptions associated with the equations that we're writing down, so things like chemical reaction rates, like the Michaelis-Menten kinetics and the decay term. And so we take what's called a model-independent approach, and this isn't new for our project. It's been around for several decades, but essentially we're using something about the structure of the equations rather than specific equations themselves to figure out something about the model, and we're going to see if we can create similar predictions to what we've seen in the literature without writing down equations. And so for this system, the key to doing a model-independent approach is to recognize that the cell communication network can be represented with something called a regular network. So here's an example of a regular network on the right, and it's defined as follows. So in a regular network, each cell has to have the same type. So this is realistic in our system. We also have all the arrow types being the same, indicating that the cells are communicating in the same way. And then all the cells are going to receive the same number of input arrows. So in our cell array, this means the cells are getting the same amount of signal from their neighbors. And with every regular network, we can associate a class of ODEs called admissible ODEs. So we're not writing down a specific equation, but this class has a form that's associated with the regular network. And the way you do that is as follows. You first associate with each node a state variable. So in this case, x1, x2, x3. And this can be in any dimension we want. So say we're keeping track of 4 chemicals inside each cell, then all of these are going to be in R4, so for 4 real numbers. And then we want to define the dynamics of each cell. So up here on the first line, we're defining the dynamics of x1 with respect to time. And by convention, we're always going to assume that this depends on that cell itself. So it's a function of x1 in that first argument. And then we assume it's also influenced by cells that have arrows into x1. So since over here there's an arrow from 1 to 1, I put an x1 in the second argument. And then there's an arrow from 2 to 1, so I put an x2 in that third argument there. And then the last assumption is that the cell can't distinguish between inputs from its different neighbors. And so we have this overline here that's just indicating that this function should be symmetric in those 2 arguments. And then lastly, we are going to do some what we call bifurcation theory. So we have this bifurcation parameter lambda. In our system, this represents the amount of external signal. And so we're going to vary this parameter and see if we can get new steady states associated with different patterns. And the really nice thing about this model-independent theory is that this function f can truly be anything we want, any smooth vector field. But we do have to assume that the function f is the same for all of these cells. So that's really important to our analysis. But amazingly, if we assume this, then the regular network structure is already going to restrict the possible patterns that we can see in this system. And in particular, for an admissible ODE, typically we get bifurcations that correspond to patterns. So it's a very high-dimensional system, but here schematically, we can write down a bifurcation diagram. And the way to read this is as you go from left to right, you're thinking about increasing the amount of external signal. So for low amounts of external signal, I'm looking at a slice over here, and there's just one steady state corresponding to this branch here, and that's the homogeneous steady state where all the cells are the same. Then as I increase the amount of external signal, so say somewhere like here, now I have 2 solutions. One is associated with that nice checkerboard pattern there, and then one is associated with the homogeneous solution. And you can continue to do this. As you continue to increase the amount of external signal, it seems like there are more and more patterns that can form. So this is really nice on one hand because we're able to enumerate the possible patterns, but it still doesn't quite match what we see in biology because oftentimes in development, there's one pattern or maybe a small subset of similar patterns that are realized every time in development. And so what we use is the fact that only the first bifurcating solution near this homogeneous steady state is going to be stable. And then we ask ourselves the question, what additional assumptions do we need on the system to predict the pattern of this first steady state, which tends to be like the globally stable solution that is realized in a real system? And what we find is that if we add some qualitative features of the chemical signaling network in addition to the cell communication network, we can predict the pattern associated with this first bifurcating pattern. So this is an example where we have a 16 by 16 array of cells with periodic boundary conditions. They're communicating with their neighbors and next nearest neighbors as shown on this bottom scheme here. And then we're assuming qualitatively that we have this coupling scheme. And when I say qualitatively, we're really not assuming anything about the kinetics, so no mass action kinetics, no Michaelis-Menten kinetics. We're just assuming that Notch is inhibiting delta within a cell, and then delta is activating Notch in neighboring cells. This tells us some information about the sign of certain derivatives, and from that we predict the pattern associated with that first bifurcation. And this matches the simulations and a lot of the things that we see in the literature. Also interestingly, if you want to change the pattern, you can see here that there are kind of 2 ways to do that. Either we could change the cell communication network on the bottom there, or we could change the chemical signaling network. And so again, this matches what we see in the literature. If we allow the cells to communicate over longer distances, as shown in the bottom here, then we can get some coarser grained patterns like stripes that span over multiple cell widths. Or secondly, if we don't change the cell communication network, but we observe a coarse grained pattern like shown on the left, then our theory implies that we must have additional connections, so more chemical signaling in the cell. And in particular, we must have either this on the top or this on the bottom. Okay, so this is really nice. We've been able to predict a lot of the things that we see in the literature without making assumptions about the chemical reaction rates. So the next natural question is, can we generalize this beyond Notch signaling? And to do this, we need to get a little bit more into the math. And so we're going to try to split the system into the chemical signaling level and the cell communication level. And since we're doing bifurcation theory, a lot of this information is encoded in what we call the Jacobian. It's like a linearization or an approximation of the system. And we want to see how the chemical signaling network and the cell communication network enter this Jacobian. So a lot of the chemical signaling dynamics is kind of encapsulated in this function f. And since by convention, how a cell influences itself enters the first argument, if we want to understand how what we call the internal dynamics, how a cell influences itself, we take the derivative with respect to the first argument or the differential, and we're going to denote this with a q typically. And then similarly, how a cell is influenced by its neighbors enters this function f in the second or third argument. And so if we take the differential with respect to the second or third argument, we would get what we call the coupled dynamics, and we denote this with an r. And since this function f is symmetric in those arguments, it really doesn't matter if we take the derivative with respect to the second or third argument. If we're evaluating at the homogeneous steady state, that'll give us the same thing. Okay, so then on the cell communication level, we can encode this information in the regular network in what's called an adjacency matrix. And the way to get this is as follows. We first just go row by row, and row 1, I'm looking at the inputs into cell 1. So row 1, column 1, since there's this arrow from 1 to 1, I put a 1 there. And then row 1, column 2, since there's an arrow from 2 to 1, I put a 1 there. You just continue like this. And what you'll see is that when you take the Jacobian, you get this really nice structure where I can decompose it into the sum of this matrix, which depends solely on the internal dynamics, and then this matrix, which has r's, so it depends solely on the coupled dynamics, but the r's are in the place of the adjacency matrix. So if q and r are just scalars, I could write this as q times the identity matrix plus r times the adjacency matrix. But since in general q and r can be matrices themselves, I just have to use this fancy Kronecker product here. So we have q Kronecker product identity plus r Kronecker product A. And when you recognize this structure, you get this really nice theorem due to Golubitzky and Lauterbach in 2009. It's a little bit long, so this one I'm just going to read, and then I'm going to talk about why I think it's important. So it says that the critical eigenvalues of the Jacobian evaluated at a synchronous steady state are the union of the eigenvalues of q plus mu I r, where q and r are the internal and coupled dynamics respectively, and mu I is an eigenvalue of A. The eigenvectors are u Kronecker product vi, where u is an eigenvector of q plus mu I r, and mu I vi is an eigenvector pair of A. Okay, so a couple nice things. The first is that these are typically very high-dimensional systems, and so looking at the Jacobian, that's like a huge Jacobian. So this is telling us that instead of looking at that, we can look at many smaller matrices, q plus mu I r. The second really nice thing is you can see where the chemical signaling dynamics and the cell communication dynamics enter these matrices. So there's clearly these q's and r's, which depend on the cell level dynamics, and then the mu sub I, which depends on the global cell communication. And then lastly, when we're doing bifurcation theory, the critical eigenvector is going to tell us a lot about this bifurcating pattern. And so we're able to split this critical eigenvector into two parts. We have this part u, which intuitively is like what's happening within each cell, and then this part vi, which intuitively is what's happening on the global level. And since we care about the global pattern between cells, when we look at this vi, this is going to tell us what the bifurcating pattern is. So our addition to this theory is we take this theorem and we're able to create criteria for these different patterns to form when there's two chemicals inside each cell. And to create this theorem, we need a few assumptions. So a first assumption is that this adjacency matrix A has real eigenvalues mu1 through mu k with algebraic multiplicities alpha 1 through alpha k. And we think this is a somewhat realistic assumption because in many of these cell communication networks, the adjacency matrix is going to be symmetric, so that will give us real eigenvalues. We also assume that the determinant of R, the coupled dynamics, is 0. And again, this is somewhat realistic. It matches a lot of the modeling that we've seen in the literature, and it reflects something about the sparsity of chemical signaling between cells compared to within cells. So again, that's somewhat realistic. And then lastly, we define this quantity B. It's the trace Q, trace R minus trace QR. Where that's coming from isn't too important, but I want to point out that this does depend on the chemical signaling network solely, and this is going to enter the criteria. So in our preprint, we have 12 possible bifurcations, but here I'm just going to give the 4 generic bifurcations. And when I say generic, I mean that under an arbitrarily small perturbation of the other bifurcations, you can get to these cases. And so in biology, which is very noisy, these are the most likely to occur. And in the rightmost 2 columns, we give conditions on the chemical signaling network. And then on the very left, we're given something that kind of tells us how to compute the pattern. And what I would take away from this is that the first 2 rows tell us that the pattern is going to be associated with the eigenvector associated with the smallest eigenvalue of the adjacency matrix. And then these second 2 critical pattern spaces, no pattern will form. A little bit more information. So the first row and third row are going to be steady state bifurcations, so they lead to a steady pattern. And then the second and fourth row are going to be Hopf bifurcations, meaning that there will be oscillatory behaviors in the solutions. Okay, so far we've seen that we can predict a lot of the patterns in the literature by removing, and we can remove a lot of these unnecessary assumptions about chemical reaction rates. And when we remove these assumptions, we're actually in some ways able to do a little bit more complete of an analysis because we can really provide necessary and sufficient conditions for certain patterns to form. And so this leads to a few nice applications. The first is that we can predict molecular causes of pattern breakdown more readily. And then the second is that if we have some partial information about the chemical signaling and then we know something about the pattern, we can predict more information about the chemical kinetics. So in the first application, I want us to imagine a scenario. Maybe there's an experimentalist. They're looking at a model organism, so like C. elegans, Drosophila, and they're looking at some pattern formation in the system. And they know that the chemical signaling is due to a pathway like this. So I know that I said for the theorem we could only do it for 2 chemicals inside each cell, but we can actually generalize it as long as the coupling between cells satisfies some technical conditions. Okay, so if I'm the experimentalist, maybe I want to know what the molecular causes of breaking the pattern are. So I add some perturbations, I want to see when the pattern breaks. And I think some of them are more obvious. So for example, if I knock out this arrow from 4 to 5, then I'm breaking this whole chain. So it makes sense that the pattern will break. Same for 6 to 7. But I claim for our theory, we can find some less intuitive ways that the pattern can break. So for example, our theory also says that if we make this arrow from 1 to B very small, we could also break the pattern. And then we can also infer more information about chemical kinetics if we know partial information about the chemical signaling and the pattern. So I have 3 examples here. So in the first example, again, we're in an array of cells, but each cell is communicating with its neighbor via one chemical influencing another chemical in the neighboring cells. In this case, we can only see steady state bifurcations, and we can't see oscillatory behavior. So that's what this no hop thing means here. But if we see a pattern, that means that the U's have to inhibit each other. So we're getting a little bit more information about the system. In the second case, we have a similar thing. U is interacting with U in neighboring cells, but U is also interacting with another chemical V within each cell. In this case, we can get a range of patterns. So we can get synchronous oscillations, in which case we would have to have the U's activating each other, or we could see an oscillating pattern or a steady state pattern. And in both of these cases, the U's would have to be inhibiting each other. Okay, and then the last case, most similar to notch signaling, we have U interacting with another chemical V within a cell, and then U is influencing V in neighboring cells. So in this case, we also cannot see oscillatory behavior, so no Hopf bifurcations. But if we see a steady state bifurcation, there's actually 2 ways that we could see that behavior. The first is like we see in the model, where V is inhibiting U within a cell, and then U is activating V in neighboring cells. But we could also switch the activation and inhibition. So we could have V activating U within a cell, and then U inhibiting V in neighboring cells. So I think, obviously, this is very simplified. We're looking at just 2 chemicals inside each cell. But I think for me, this brings up an important point because in mathematical biology, it's often very tempting to write down a model, say, look, this model fits our data, and so this model must say something mechanistic about the system. But I think that's a little bit of a fallacy because what this is showing is that if the qualitative behavior that we care about is the pattern, there's often multiple models that can create that. And so I think one of the most powerful things about this model-independent approach is that we can really create some minimal conditions that give us the qualitative behavior that we're observing in our data. So I think that's very powerful. And I know that's a lot of theory, but I like to think of it like this. So we have these 3 things interacting. We have the cell communication network, the chemical signaling network, and the pattern. So we started by showing that if we have the cell communication network and the chemical signaling network, we can predict the pattern. But again, since we have those necessary and sufficient conditions, if we have 2 of these things, we can predict some information about the third thing. So if I have the chemical signaling network and the pattern, I can predict something about the cell communication. Or if I have the cell communication and the pattern, I can predict something about the chemical signaling. Okay, so in conclusion, we started by using a model-independent approach to study these patterns we've seen in the literature. And we saw that we can predict many of these patterns by removing assumptions about chemical reaction rates. We were also able to do a slightly more complete analysis of this pattern formation, and this led to some nice applications. So we showed that first, we can more readily infer unintuitive causes of pattern breakdown. And then second, we showed that if we have partial information about the chemical signaling mechanism and we know the pattern, we can infer more information about the chemical kinetics. Of course, there's a lot of future work. So the first is that I said that we have to make some requirements or some assumptions about the chemical signaling between cells. And so in the future, we'd like to remove this assumption and be able to do it for arbitrarily many chemical signaling networks. And then the second, I think I've tried to convince you throughout this talk that we're making fewer assumptions, but in some senses, we're making actually a lot more assumptions about the cell communication network because we require that it's regular. So we're assuming all the cells are identical, they're communicating in exactly the same way, and we're assuming that this adjacency matrix has real eigenvalues. So this is a huge assumption, and it's actually probably never satisfied in most biological systems. So a key next step is to incorporate small perturbations to this network structure and see if these theoretical results will hold approximately. And then I'd like to end with some acknowledgements. So first, I'd like to acknowledge my lab group, the Dawes Lab at OSU, with our principal investigator, Dr. Adriana T. Dawes. But yeah, my lab members, they always have great suggestions, so thanks to them. And then some other collaborators at Ohio State, Dr. Marty Golubitsky in the math department at OSU. He's one of the pioneers of this model-independent approach for networks. And then Dr. Helen Chamberlain in the Department of Molecular Genetics at OSU had some very useful suggestions about the applicability to biology. Sorry, let me go back. Okay, so most of this is in this preprint titled here. It's in submission at the Journal of Mathematical Biology. Hopefully it'll be out soon. And at this point, I'm happy to take any questions. Thank you.
Wonderful talk. Thank you so much. I have a question regarding the lattices you showed. So you have the square lattice or hexagonal lattices. So can you comment on the lattice type changes? Can you see in your theory how maybe your patterns change under otherwise more or less identical conditions?
Okay, so it's not obvious to me from the lattice itself, but I think the key thing is that the lattice changes the eigenvectors of the adjacency matrix. And so in our theory here, sorry, go back. Okay, so here, essentially the pattern is corresponding to the eigenvectors of that adjacency matrix. I haven't seen that much theory about how the lattice affects those eigenvectors, but that's definitely something I'd be interested in if you know anything about that. Does that answer your question? Or yeah, okay, thanks.
Hey, nice talk. Quick question. You might mention this, but I assume that your assumption here is for linear systems and also for what happens with higher order. If you have higher order dynamics, is this something that you plan on pursuing in the future?
It's a good question. So I think actually one of the strengths of this theory is that if I go back to the admissible ODE, so there's no assumptions being made on this F other than that it's a smooth vector field. So it can actually be any order that we want. The key though is that the coupling between cells has to be sparse. So like the chemical signaling between cells has to be sparse. So that simplifies our computations a little bit, but you can actually have as many nonlinearities as you want in this function F. Yeah, thanks.
Thanks for the talk, Liam. Have you thought about doing this on a 3D lattice?
Thanks, Rob. That's a good question. I have not thought about doing this on a 3D lattice, but that's definitely an interesting direction to go. And the theory should apply pretty directly. You just have to order the cells a little bit differently for your adjacency matrix, but everything should work out okay.
Hi, thanks for the talk. I was wondering if you could extend this to off-lattice systems at all?
Yeah, so you can extend it to off-lattice systems. The key is that you just need a regular network here. So for example, if I truly had cells communicating like this, I could do this network right here. And I think it's nice, like there's not even any symmetry in this network. It's just the regular network structure. As long as you have that regular network structure, everything should work okay.
Thanks for the talk. So in one slide, you mentioned that if you assume you know partial knowledge about the chemical reaction network, you can infer something about interactions. I'm wondering how much of that can you take away? Like if you assume it's a complete network, which means you know nothing about it, how much can you say in that case?
Yeah, that's a great question. So what we're doing when we do those computations is we're using the criteria, and the criteria tend to depend on like the trace and the determinant of certain matrices. And so when I'm making those partial assumptions, it's about which chemicals are influencing each other. That allows me to set certain elements in those matrices to 0. And so that's really helpful with the calculations. But definitely the more chemicals you incorporate, there's going to be a larger class of systems that can create the same dynamics. And if you make fewer assumptions about which chemicals are communicating, again, you're going to have a larger class of systems that create the same dynamics. Yeah, thanks for the question.